Okay, so we're gonna start with chapter 17, and that's the endocrine system. So what you should be able to tell me about endocrine system is you should be able to compare the endocrine system and the nervous system. You should be able to list what are the similar things between these two systems, and then what are the differences. So overall, you would basically just read that section there and be able to tell the similarities and differences. And that's that. So how do they act on tissues? What do they act on? Things like that. From that 17.1. Table 17.1 is a good kind of a summary of the differences between the endocrine and nervous system. So again, be able to tell me, what is the communication method for each, response time, the effect, the duration, how long does each last? So table 17.1 is a good one for that. Endocrine glands, you should be definitely able to divide the endocrine glands into the major endocrine glands and then just organs or tissues with endocrine function. And for this, if you have done your endocrine system lab exercise, you've already done this. You have that table, that first table, you have the major endocrine glands, and then on the right side, you have the other structures. And figure 17.2 shows you this, this division here. So you have on the left side, you have the major endocrine glands. On the right side, you have the organs or tissues. So be able to divide them, be able to categorize them, okay? Be able to tell me how we can stimulate endocrine system. So there are 3 ways of stimulation. You have the hormonal stimulation, humoral stimulation, and nervous system stimulation. First, memorize what are the 3 types, and then go deeper and make sure that you know what each of these means. Okay, so that's stimulation. Then we have that infamous table 17.2. It's a huge table with all the organs and structures that produce hormones. And it also has all the hormones and what these hormones are acting on, what they do, what's the function. You basically, I wouldn't memorize the table. As you go over each structure further in the chapter, you are learning this. So table 17.2 just summarizes everything for you, but you can take it one gland at a time or one organ at a time, and you will eventually basically learn that table 17.2. So that's that. Categories of hormones. Be aware of the 2 major categories of hormones, which are the circulating hormones and local hormones. Circulating hormones we divide into the 3 groups. You have the protein hormones, biogenic amines, and steroid hormones. So those are the 3 categories of the circulating hormones. And then we have the local hormones, and local hormones sometimes are also known as just the signaling molecules. And know for this that the main group of local hormones are eicosanoids. Okay, so that's that. I would like you to know also that the steroid hormones are lipid soluble, they're not water soluble. And then the biogenic amines and protein hormones are water soluble. This is going to be important when we look at the transport and interaction of the hormones with the cells. There is one exception to biogenic amine, and that's thyroid hormone. It is not water soluble, it is lipid soluble. So that's the hormones. Okay, local hormones, eicosanoids, know that name. And then the biggest group of eicosanoids are prostaglandins. So that would be beneficial to know as well. So you don't have to tell me what prostaglandins are doing, just know that that's where they belong, okay? Hormone transport and hormone interactions with target cells are going to be dependent on solubility. So as I said just a moment ago, you have the steroid hormones that are lipid soluble, and you have the protein hormones and biogenic amines are water soluble. But the water soluble hormones simply can dissolve in blood plasma. Remember, hormones travel through blood, right? So water soluble hormones can simply dissolve in plasma, in blood plasma, which is mostly water. But the steroid hormones, because they're lipid soluble, these are not able to dissolve in the blood plasma. And so what they have to do is they have to get a carrier protein. So a carrier protein or protein carrier, whichever way you want to call it, is kind of like a boat for the hormone, and it allows it to travel in the blood plasma. So that's what's necessary for the lipid soluble hormones. Now, often the water soluble hormones, even though they don't require a protein carrier, they often do jump onto a protein carrier because it prevents them from degrading too fast. So it provides protection for these hormones. So that's hormone transport or transport. The interactions of hormones with target cells are also going to be dependent on solubility. And this has to do with the fact that cell is, the boundary of a cell is a cell membrane, and cell membrane is a lipid bilayer. So when a lipid hormone comes to the cell membrane, it can simply pass through that lipid bilayer and enter the cell. And so a lipid soluble hormone will interact with intracellular receptors. Okay, so it will interact with receptors that are on the inside of the cell. But the water soluble hormones, these cannot pass through the cell membrane. Cell membrane will repel them. And so water soluble hormones have to act with receptors that are sticking to the outside of the cell. And we call these water soluble hormones as first messengers. Okay, I would like you to be able to tell me the cascade of the events that happens when a lipid soluble hormone interacts with the cell versus when a water soluble hormone interacts with the cell. I would suggest you create a flow chart. Okay, so you can, and you can just look at the image. I like to work with pictures that are in the textbook. Look at the picture and trace it on that picture. Okay, you would have the first thing you would have is hormone unbounds or unbinds from its carrier protein in the blood so that it can exit the blood. So hormones do not carry their boat with it, right? They're going to leave this in the blood, and then the hormone exits the blood vessel and it's going to come to a target cell. This lipid soluble hormone will be able to pass through the cell membrane. And once it's in the cell, inside of the cell, it will bind to a receptor on the inside of the cell in the cytoplasm. The binding will then form a hormone receptor complex. Okay, this hormone receptor complex will travel to nucleus, and in the nucleus, it will bind to hormone response element. Hormone response element is an area of DNA in the nucleus that is specific for this hormone receptor complex. So there's like this specific match that is able to happen inside of the nucleus. Once this hormone receptor complex binds to the hormone response element, and see there, these terms kind of sound similar, so make sure that you write them all out and memorize them that way. So once this binding happens to the DNA, this initiates transcription of DNA. And transcription means that we're rewriting DNA code into mRNA. Okay, this mRNA will eventually exit the nucleus, and in the nucleus, mRNA gets translated into a protein. And then this protein does something, okay? And this is going to depend on what hormone this was, okay? So this protein will, in any case, the protein will either alter the shape of the cell or the function of the cell, okay? And that's going to change what that cell is doing, okay? So that's the lipid soluble hormones. For water soluble hormones, this is a little more complicated. It has two ways of going about it, but in each case, the hormone, the water soluble hormone, acts as a first messenger on that receptor that's sticking to the outside. This initiates activation of G protein, and then the G protein can either interact with adenylate cyclase or it can interact with phospholipase C. Okay, both of these pathways is going to result in activated protein kinases, and kinases are molecules that phosphorylate other molecules. Phosphorylation of other molecules will either activate or deactivate those molecules. So again, it depends on what molecule specifically we are talking about, but it has capability of either activating something or deactivating something. And phosphorylating means adding a phosphate group, chemically speaking. The phospholipase C is the more complicated pathway here, and the phospholipase C also affects activity of ion channels. Whereas adenylate cyclase just results in activated kinases. Phospholipase C will give you activated protein kinases, and it will affect ion channel function. You don't have to tell me the intermediate steps there, like the GTP, the DAG and PIP2 and IP3 and all that. Okay, you don't have to know that. I just kind of want you to know the big picture here for those. The one section that I kind of skipped over because it's between the transport and the interaction with cells is the levels of circulating hormone. So be able to tell me how are the levels of circulating hormone changing. So we can increase hormone in our body by producing hormone. So your glands will release extra hormone and that will increase your hormone levels. And then the decrease in circulating hormone levels will be due to hormone elimination. And you can eliminate hormone either by cells using it or interacting with that hormone, or your kidneys could eliminate this hormone, or you can just break it down in your liver. Okay, so several ways that you can eliminate hormone from your body. Okay, so what else? The degree of cellular response, be able to tell me what is upregulation and what is downregulation. So if your body is low on certain hormone, but your cells need to respond to it, they will upregulate their receptors, meaning that they will show more or they will express more receptors either on their cell membrane or inside of their cytoplasm, so that they're more likely to capture the few hormone molecules that are out there. Now, if there is too much hormone, then the cell can do the opposite and it can downregulate its receptors. And this means that it will deactivate some receptors, so that as the cell is getting overloaded with all these hormone molecules, there's only a few receptors that can interact with it, so it's not overstimulating the cell. So that's upregulation, downregulation. Be able to tell me what are the three hormone interactions, so the synergistic, permissive, and antagonistic, and know an example for these. So make sure that you look at that. Then we're going specifically into hypothalamus and then pituitary. So hypothalamus and pituitary are very closely related. Hypothalamus is not a major endocrine gland, but it is a very, very important player or part of the endocrine system. Hypothalamus releases 5 releasing hormones. It releases 2 inhibiting hormones, and then it releases 2 hormones that will be passed on to the posterior pituitary. This should be reminding you of that big table in your lab exercise. And so when you're doing these lab exercises, it's basically like your notes. You are putting the information together, and that helps you study. So take out that lab exercise, and first make sure that I haven't left any comments for you on my TCC. And if I have, read them. And then if everything was good, then you can take that lab exercise and study that. Okay, so hypothalamus releases 5 releasing hormones and 2 inhibiting hormones, and 2 of those that go to posterior pituitary. Be able to list them and be able to tell me what these releasing hormones release. Okay, all the releasing hormones release something from anterior pituitary. So you need to be able to tell me what they release. And then the hormone from the anterior pituitary has a certain function as well, which, if you're recalling the table and the lab exercise, that would be the 3rd column. And so be able to tell me the function of that anterior pituitary hormone as well. The inhibiting hormones are going to inhibit the release of prolactin and growth hormone, because the 2 inhibiting hormones are growth hormone inhibiting hormone and prolactin inhibiting hormone. And I had several people say that they inhibit prolactin from hypothalamus and growth hormone from hypothalamus. Well, those two don't come from hypothalamus. They still come from the anterior pituitary. So the inhibiting hormones also inhibit hormones from anterior pituitary. And then the last two, oxytocin and ADH, be able to match them to their nuclei. So oxytocin comes from a paraventricular nucleus, and ADH comes from the supraoptic nucleus, right? And it's just the region of the hypothalamus that produces these two hormones. And then these two hormones are going to travel to posterior pituitary. But they will be stored in the posterior pituitary until posterior pituitary is stimulated to release them. And yeah, posterior pituitary does not make its own hormones. It only takes hormones from hypothalamus and releases them when it's told to do so. Anterior pituitary does make its hormones, and it will release most of anterior pituitary hormones as a response to the releasing hormones from hypothalamus. Posterior pituitary is also known as neural hypophysis, and anterior pituitary is known as the adenohypophysis. The posterior pituitary communicates with hypothalamus by means of neurons there, and anterior pituitary communicates with hypothalamus by means of hypothalamo-hypophyseal portal system. And remember portal system, I will ask you about it in the next unit, okay, where we talk about blood vessels. But basically, it's your two capillary beds connected by the portal veins there, hypophyseal portal veins. And so that's how hypothalamus and anterior pituitary communicate with each other. Be able to label this. You should have a drawing in your lab manual exercise, so you should know what is what there. So make sure you are able to label these. The next, I'm looking at figure 1712 now, and this is where you have all those hormones that are from the hypothalamus. Okay, you have the releasing and inhibiting hormones, and then you have the anterior pituitary hormones there as well, and it tells you what it does. Below that figure, you have it written out. Okay, so there it is for you again. Make sure that you really refer to that figure and go over this several times. Okay, it's not enough to just write out the table, fill out the table and put it aside. Okay, you have to study this material. You have to commit it to your memory without looking at it, right? Because you can't use that on the exam. So make sure that you're studying this and committing it to your memory. For the anterior pituitary hormone, the representative hormone is the growth hormone. So you can just look at the chart, look at that table on page 682. This is figure 1713. And it starts at the top with the stimulus, like what is the stimulus for the growth hormone? So read that. I think that's for growth hormone, that kind of makes sense. What the stimulus would be here. You would have the negative feedback, age, time of the day, nutrient levels, stress and exercise, all that is going to be responsible for growth hormone release. And then it shows you what it does. So some of the growth hormone goes into blood directly and goes and acts on the cells and on tissues. But some of the growth hormone is going to go to liver and release insulin-like growth factor. This is going to have the same effect as growth hormone, but insulin-like growth factor has a longer half-life. So that means that it lasts longer in your body, has a prolonged effect. Look at the figure 1714 and see how your growth hormone would be affected. So you have this, if you've watched the lesson already, this is just a review. So I'm not going into everything into detail, right? So make sure that you review that. Thyroid gland, be able to tell me that how the thyroid gland is organized. So it's a bilobed gland. The middle part is isthmus that connects the two parts. And the thyroid gland has two types of cells, follicular cells and parafollicular cells. Follicular cells are the ones that give you T3 and T4. These are the two versions of thyroid hormone. And parafollicular cells give you calcitonin. Okay, so be aware of that. We have, be able to tell me what the effect of thyroid hormone is. If you look at page 687, figure 1717, Then you have the diagram for thyroid hormone. So again, stimulus and then pathway, where does it go? What does it do? At the bottom is the effector response. So you see what it does in the end. You don't really need to focus much on figure 1716. That shows you formation of T3 and T4. I'd like you to be aware that without iodine, you would not be able to make thyroid hormone. And so iodine is very important part of our diet. And we get it from salt. So when you buy salt, then just regular salt, it's iodized. Okay, so it has iodine added to it. And that's so that we are producing sufficient amount of thyroid hormone if our thyroid gland is healthy, right? The T3 and T4 are different from each other by number of iodines. So T4 has 4 iodines, T3 has 3, but I'm not going to really be asking you that much detail there, but know that iodine is absolutely necessary for your body to produce thyroid hormone. I would look at the clinical view, hyperthyroidism and hypothyroidism, and read through that. And you can see the, Graves' disease, you can see the person developing goiter. And that's basically the reason why they have the goiter is because the thyroid gland grew. It's because it was trying to meet the demand of the body for the thyroid hormone. But because probably the person was deficient in iodine, the thyroid gland couldn't make that thyroid hormone. And so it tried to grow thinking like, well, not that the gland thinks, right? But it grew so that it would try to meet that demand, but it still wouldn't work if you're deficient in iodine. So that's the clinical view. Calcitonin, there's just a little small section about that. Just know one sentence about what calcitonin does. So whatever is in your lab exercise should be enough there. Okay, next are adrenal glands. Be able to tell me what hormones come from what. So again, the lab exercise comes in handy. We have adrenal cortex, which has 3 zonas: zona glomerulosa, zona fasciculata, and zona reticularis. And they release mineralocorticoids, glucocorticoids, and gonadocorticoids, respectively, in that same order as I listed them. So be able to tell me which zona releases what. The representative hormone for the adrenal gland is cortisol, and that table is, or that figure is figure 1719. So look at that and be able to describe what is the pathway for the action of cortisol. Okay, what is the stimulus? Where does it go? What does it act on? What does it do to those organs? The adrenal medulla is releasing epinephrine and norepinephrine, and those you've already talked about in AMP1, so that we don't really talk about them here anymore. Just know that those are the ones that come from adrenal medulla. And so that's cortisol. And the pancreas is, I think, the last one, and then we just have those some organs at the end. So pancreas is a heterocrine gland. It is not the major endocrine gland. Okay, pancreas is not, but we do talk about it still. Pancreas is a heterocrine gland, which means that it has endocrine and exocrine function. The 2 hormones that come from the pancreas are insulin and glucagon. Know a little bit about the organization of pancreas. It's divided into exocrine portions and endocrine portions. The endocrine portions are your pancreatic islets, and the exocrine portions are the pancreatic acini. And the pancreatic acini produce pancreatic juices, and these will go into your small intestine to help with digestion. But the endocrine parts, the alpha and beta cells that produce glucagon and insulin, these hormones will go into your blood. Okay, so know what insulin does, and I think we have a glucagon here as well. So there's the diagram is shorter for insulin, glucagon here. So that's figure 1722 and 1723 is for insulin and glucagon. You can go ahead and read the clinical view on diabetes. So there's diabetes discussed there briefly. Okay, it's not everything about diabetes, but it's a very brief kind of a look at what it is. And the rest of the chapter, the other endocrine glands, just know what each gland releases. Okay, so pineal gland is releasing melatonin and it affects your circadian rhythm, right? The parathyroid glands release parathyroid hormone. And thymic hormones. Heart is going to release atrial natriuretic peptide. We'll talk about it in next chapter. Chapter 19 is going to talk about this. Kidneys, erythropoietin. Erythropoietin we've talked about in chapter 18. Okay, this is what stimulates formation of erythrocytes. Liver is going to produce your angiotensinogen, which ultimately will be converted into angiotensin 2. Stomach and small intestine. Stomach produces gastrin, small intestine produces cholecystokinin and secretin. Gastrin is going to affect motility of your digestive system. Cholecystokinin and secretin both will increase secretions from accessory digestive organs, so like from liver and gallbladder and pancreas. Skin is going to produce calcitriol. Adipose connective tissue produces leptin, and that's that. And then the aging and endocrine system, our endocrine system becomes less efficient as we get older, is the summary of that section. So that's chapter 17. Okay, any questions from 17 so far? I know I'm going fast, but that's just because I want to point out like the most important things. You have more detail in the lessons, recorded lessons. Okay, think about it. If you have a question, I'm going to go ahead and jump into chapter 18. So for chapter 18, be aware that the blood is part, or it travels through the cardiovascular system. So we kind of, it's actually as included as part of cardiovascular system. Blood is fluid connective tissue. Okay, so where you had the blood lab handout or the blood lab exercise, in that there was that table that said where are antigens and antibodies found, name the tissue. Well, they're found in blood. Blood is the tissue where you find antigens and antibodies when we're talking about blood typing, right? And then specifically, we had antigens on the red blood cells and antibodies on or in plasma. Blood is composed of two main parts, formed elements and plasma. Formed elements are the solid parts. Plasma is the fluid portion. Be able to tell me what are the solid parts? What are the types of cells we see in blood? And then be able to tell me the composition of plasma. Be able to list the characteristics of blood. So know what the pH should be. What is the color? What is the viscosity, the average volume? Be able to list that. That's summarized for you in table 18.1. So you can look at that. For figure 18.1, where you look at the parts of the blood, the centrifuge blood, be able to tell me the major components of each part. I don't want you to tell me the percentages. You don't need to tell me the exact percentages, but I would like you to know that plasma is mostly water. Okay. I would like you to know that out of the plasma proteins, the most abundant proteins are albumins. Okay. I'd like you to know for the Buffy coat that this is going to be your platelets and leukocytes. What is the most abundant leukocyte? Well, it's neutrophils. Okay, so be able to tell me that. You don't have to tell me it's 50 to 70%, but know that it's neutrophils that are most abundant. And then erythrocytes is the rest of the centrifuge blood, and erythrocytes would be the most abundant formed element. Okay, this question always throws off students. Okay, if I ask what is the most abundant formed element, well, formed elements are the blue and the red box on that chart, right? The formed elements are the Buffy coat and erythrocytes, right? Leukocytes, platelets, erythrocytes. Well, the most numerous formed element would obviously be erythrocytes. It's almost half of the blood. Now, buffy coat is a very small portion of a centrifuge blood, so none of those could be the most abundant, right? And most abundant means what do you have the most of, okay? Like as far as numbers go. Know what a blood smear looks like. I will not ask you to tell me, what is this? Is this a neutrophil or whatever? So you don't have to memorize that, but be able to tell me what, just the composition of blood plasma, composition of the buffy coat, and then a little bit about erythrocytes. So you have a bunch of tables here. I'm not going to ask you about the common electrolytes in plasma and their values. So you don't need to do that. So if I ask you a number, if I want you to know a certain number, I will tell you that I want you to know this specific number. Okay. These tables that you see here are going to be basically for your reference only. Okay. Let's look at the hemopoiesis. Hemopoiesis, the figure 18.3 is very useful for you. So you can track what is being formed, how, by what process. Know what cell hemopoiesis begins with, and that is hemocytoblast. Know where hemopoiesis happens. It happens in red bone marrow of certain bones. But this also depends on development, as I mentioned in the lesson. During embryonic development or early fetal development where there are no bones yet, the hemopoiesis would happen in the yolk sac and then later in the liver and spleen before the red bone marrow forms, right? But in adults, it's going to be in the red bone marrow of certain bones. The hemopoiesis, the hemocytoblast can be stimulated to either enter the myeloid line, which is like a process, or a lymphoid line, which is another process. The lymphoid line will only go through the process of leukopoiesis, and it will produce lymphocytes. So it will produce your B lymphocytes, T lymphocytes, and natural killer cells, which are also lymphocytes, but it doesn't have it in their name. The myeloid line can go through the process of erythropoiesis, thrombopoiesis, as well as leukopoiesis. Okay, so be able to tell me what does leukopoiesis from myeloid line produce? And it will form, if you look at the bottom, okay, it's all the phils, so all the granulocytes, so eosinophils, basophils, and neutrophils, and then the monocytes. And monocytes become macrophages ultimately. The myeloid line can then go through the process of thrombopoiesis, and thrombopoiesis is formation of platelets. If you flip to the next page, if you have a hard copy of the textbook, if you don't, if you scroll down below that table, you will see figure 18.4 that shows you platelet formation. So how do the platelets form? And it's basically your megakaryocyte, those proplatelets get chopped off by blood flow as they protrude into the blood vessel. And then erythropoiesis is formation of erythrocytes. This is stimulated by erythropoietin, which comes from kidneys. Be able to tell me what stimulates the release of erythropoietin. That is somewhere, I think, wherever we're talking about erythrocytes. So be able to list the conditions that stimulates release of erythropoietin. And then be able to tell me the general changes that the cell goes through as it's on its way to become erythrocyte. So you don't have to tell me each individual step and each name on that flow, but know that the cell will be stimulated to produce more and more hemoglobin. And that's why it's becoming more and more red. And ultimately, it will eject its organelles, and it will eject its nucleus as well. And then in the end, you will have this biconcave disc kind of a structure, which is the final erythrocyte. Some general information about the erythrocyte. So I'm not going to ask you like its size or anything like that, but know about how long on average does an average erythrocyte live. And that's about 120 days. Be able to tell me the structure of the hemoglobin. So know that it's 2 alpha chains and 2 beta chains, and each chain has that heme group with a porphyrin ring, and inside is the iron atom. the iron atom or iron ion is where the oxygen binds to, okay? So when you look at the picture of hemoglobin, you have those four chains, four parts, right? Those four wiggly kind of parts. And each of those parts has one iron atom. So the whole hemoglobin, one hemoglobin molecule can carry four oxygen molecules, okay? Hemoglobin can also carry carbon dioxide, and hemoglobin can also carry hydrogen ion, but they don't attach to the iron, okay? They are going to attach somewhere to the protein, and we'll talk about that a little more when we talk about respiratory system. So that's that. You have figure 18.7 shows you the release of erythropoietin. So this is what tells you how or what stimulates the release of erythropoietin. So make sure that you know that. For erythrocyte destruction, know just the terminology. Again, I would make maybe a flow chart of what becomes what as it breaks down. So know the basics of that. Okay, the next section is on blood types. Be able to tell me how blood typing works. Be able to describe each blood type in terms of what antigens there are and what antibodies there are. One important thing is that what you need to realize is that if you are, I'm just going to take type A. Okay, if you're a type A person, if you have blood type A, you have A antigen on your surface of the erythrocytes, and you have anti-B antibodies. These two do not interact with each other. They coexist, but they are not a match, okay? Anti-B antibodies will not attack your A antigens. The anti-B are against B blood type. It's against the blood type that you don't have, okay? That will be foreign to you. So look at that figure 18.9 and look at, what antigens are there and what antibodies there are in each blood type. Be able to recall how blood typing was working. So that simulation, if you forgot, go back, redo it, okay? You will be asked some questions from the blood typing lab as well, from the simulation. And then we have the Rh incompatibility and pregnancy. Again, just know the basic information about that. And that is that this happens when the woman is Rh negative and the baby is Rh positive, okay? If she's Rh negative and the baby is Rh negative, then there's no problem, okay? So this is only affecting an Rh negative woman that carries an Rh positive baby, okay? That first baby, during pregnancy, nothing happens, but during childbirth, there is a slight exposure of the mother's blood or mother's body to the baby's blood and vice versa, because birth is bloody, okay? Let's be honest, it is messy. And so the mom does get exposed to the baby's blood. If the baby is Rh positive, her body gets stimulated to produce anti-Rh or anti-D antibodies. Now, that doesn't do anything to her because she doesn't have that antigen. And so, okay, fine, now she has some antibodies. What's the big deal? It doesn't harm her. Where the problem lies is if she gets pregnant with the second Rh positive baby, then these antibodies that her body formed could attack the baby and could harm the baby. And so now if she gets pregnant again and the second baby is Rh negative, that Rh negative baby is fine. Okay, it's only another Rh positive baby that's at risk, and generally first one is not at risk. So that's how that Rh incompatibility works. In table 18.7, you have the different types of leukocytes described, and so I'd like you to know just their main function. Okay, so what is the main function of neutrophil? Well, it engulfs things. Eosinophil has some like a role in allergic reaction, but it also is going to act on like multicellular parasites like worms and things like that. Basophils release heparin and histamine. Okay, you don't have to tell me anything about their nuclei. Okay, I don't care about that, but just know their major function, because in chapter 22, yeah, or 21 or 22, this is where we're going to be talking about the immune system, and this will come back. And that will be just a review at that point, because you already learned it here. So be able to tell me the function. You don't have to tell me the percentages, but know which one is the most abundant. So that's for the leukocytes. Be able to tell me how we can use the numbers of leukocytes in our body. So the differential count, what is that? And but I'm not going to ask you to match like specific conditions to a specific, like either high or low type of any of the leukocytes. So yeah, that's that. Platelets are going to be important in the hemostasis. So for hemostasis, be able to tell me the 3 steps first for hemostasis. First of all, don't get hemostasis and hemopoiesis confused. Hemopoiesis is formation of formed elements. Hemostasis is blood clotting, basically. It's how we repair things. So that's, we are stopping bleeding. Okay, stasis means stopping. So hemostasis, you're stopping blood from escaping, right, from bleeding out. So there are 3 steps: vascular spasm, platelet plug formation, and blood clotting, and be able to describe them. For the platelet plug formation, you should be able to list the function of these platelets being transformed or being morphologically changed. So as the platelets are sticking to each other, this is a positive feedback, by the way. So you have, you have the breakage in your blood vessel that exposes collagen fibers, and you're going to have platelets starting to stick to the area. As they're sticking to the area, they're releasing these chemicals, and those chemicals are attracting more platelets. So it's positive feedback. It's amplifying itself. And as these chemicals are being released, they're also prolonging your vascular spasm. They're attracting other platelets, and they stimulate coagulation and stimulate the repair of blood vessels. So be able to list that. And then we have the coagulation phase. That's our 3rd phase of hemostasis. And this is the formation of the actual blood clot. So we'll basically reinforce that platelet plug with fibrin fibers. And there's a way that this is happening. There are these clotting factors that are involved here. And if you look at figure 18.13, this gives you a very nice visual as far as how these pathways, or how this process happens. All right, so you can look at that. Intrinsic pathway is the more complicated one. It has more factors there, and intrinsic pathway is initiated by damage from inside. Extrinsic is initiated by damage from the outside. Intrinsic pathway is slower. Extrinsic pathway is much more rapid. After each intrinsic and extrinsic pathway, we get to factor 10. Okay, it looks like factor X, but it's factor 10. Okay, so after end of extrinsic and intrinsic pathway, both of these pathways gives us factor 10. And then this factor 10 goes through the common pathway. And at the end of this, we get the fibrin that will stabilize the blood clot. Then the blood clot is not going to stay there forever. It is ultimately going to promote repair of the vessel. And so imagine the blood clot is being kind of like squeezed from inside. It's called clot retraction. So there's this protein inside of platelets that is going to contract, and it's going to cause that blood clot to squeeze on itself. As it's doing that, some stuff is coming out of that, and that has some signaling properties. That's not really that important. But as that's happening, as the clot is getting smaller and smaller, it's pulling the walls of the vessel together. So it's promoting that repair, closure of that vessel. And as this is also happening, fibrin fibers will begin to break down in the process known as fibrinolysis. So we're breaking down those fibers that hold that clot together, okay? Development and aging of blood. So I've kind of mentioned that already, that there are other places where we have hemopoiesis happening in the early development, early human development. And then the blood ages, the blood cells live about 120 days. And older individuals tend to have less red bone marrow. And so the blood number of blood cells is not as high, possibly, and they're more likely to experience anemia. So that's chapter 18. So that's 2 chapters in 48 minutes. Look at that.
I think I'm going to die.
No, it's not that bad.